In the middle of the ocean, under 800 meters of pressure, swims a creature of astonishing biological complexity and market value, the Atlantic bluefin tuna. Some specimens exceed 600 kilograms, a moving muscle of pure protein, capable of sprinting underwater faster than many land animals can run. But its real race begins the moment it leaves the sea. From that instant forward, an invisible countdown starts. The challenge is not simply to transport a fish, it's to maintain molecular integrity, thermal stability, and cellular texture, all within a 24-hour window. And every misstep, every thermal delay, costs thousands of dollars in lost value. These fish are not treated as cargo. They are handled as if they were perishable medical samples. From hook to surgical processing line, every stage is calibrated down to fractions of degrees and seconds. Why? Because if a 600-kilo tuna isn't chilled to 1.5 degrees C within 30 minutes of capture, it begins to degrade. Not slowly, but exponentially. Enzymatic reactions start breaking down muscle tissue. Acidic byproducts accumulate in the blood. Color changes. Texture shifts. What could have been premium-grade sashimi becomes ordinary or rejected entirely. In Tokyo's Toyosu market, this is not a theoretical loss. It's a price drop of 20%, sometimes more, for every mistake in that narrow thermal corridor. To truly grasp the precision required, imagine a microchip factory. There, microscopic circuits are etched onto silicon wafers in clean rooms where temperature variations of even 0.3 degrees Celsius can ruin an entire production line. Tuna handling operates under a similar standard. Cooling systems on board fishing vessels use high-flow glycol loops and pressure-assisted chilling tunnels that resemble those used in semiconductor cooling. The objective is the same: immediate thermal transfer. Delay it, and structural integrity is compromised. In one case, the circuits fail. In the other, the flesh softens, the color dulls. The market says no. There's also a biological phenomenon at play, one few consumers have ever heard of: the blood acidification threshold. When a tuna is captured, the stress triggers a cascade of chemical changes. If the bleeding isn't done immediately and correctly, if blood remains trapped in the capillaries and muscle fibers, the resulting acid buildup destroys intracellular cohesion. The color darkens, water retention drops, texture becomes inconsistent. What looks like just a fish is, in fact, a ticking metabolic system that must be neutralized with surgical efficiency. Not frozen, but biologically paused. To visualize what this transport process is like, imagine the mining industry. When ore slurry is pumped through kilometers of pipeline, engineers must calculate the perfect balance between viscosity, flow rate, and temperature. If the slurry thickens or cools too quickly, it clogs. If it flows too fast, it erodes the line. Moving tuna from boat to dock to plane follows a similar physics. It's not a shipment, it's a high-value fluid system. Each crate, each transport stage, each cooling chamber is part of a pipeline where the content is muscle tissue, the risk is biochemical decay, and the margin of error is razor thin. This is the quiet genius of modern food logistics. A choreography where engines, sensors, and human judgment converge to deliver a single animal in perfect condition, 24 hours after leaving the ocean. Not by chance, but by design. Not by speed alone, but by understanding the language of heat, blood, and biology. This is not just about tuna. It's about the invisible systems that sustain what we eat, and the engineering minds behind them. 12 minutes. That's all it takes for a transformation to begin, unseen to the eye, but definitive in quality. The moment a tuna leaves the water, its muscle tissue enters a state of biological urgency. Hemoglobin begins to oxidize. Myoglobin, the protein responsible for the vibrant red color of sashimi-grade cuts, reacts with residual oxygen and iron. If exsanguination isn't performed immediately, that deep, translucent crimson becomes a dull, oxidized brown, not because it's unsafe, but because it's unappealing, less flavorful, and downgraded in the market. A delay of minutes during bleeding is enough to alter the chemical composition of the flesh. What the ocean built over years, time unravels in moments. To counter this, some vessels now operate with pressurized chilling tunnels, compact thermal chambers modeled after the blanchers used in vegetable processing. These units pull seawater through filtration membranes and inject glycol-based coolant at high velocity, surrounding the tuna with sub-zero fluid from head to tail. In less than 18 minutes, a fish that moments ago held an internal temperature of 30 degrees is brought below 2 degrees, not frozen, stabilized, below the threshold where enzymatic reactions begin to break down fiber structures. It's not simply a cooling method, it's a rapid biochemical arrest, a way to pause time, not just slow it. The level of precision required borders on the scientific. The 2023 study by Japanese marine biologists measured the impact of thermal deviations during this cooling phase. A margin of just 0.5 degrees Celsius was enough to lower the sashimi grade rating by 2 full points on the Toyosu market's sensory scale. That seemingly minor variation translated into a 35% loss in auction price. And it's not hypothetical. These errors happen when cooling tanks are overloaded, when compressors cycle unevenly, or when the chilling tunnels fail to maintain uniform pressure zones along the fish's body. It's the kind of precision normally reserved for labs. Applied here to a deck soaked in salt water and blood. Off the coast of Malta, a tuna vessel underwent a retrofit that changed the entire equation. The ship replaced its traditional slush ice tanks, where cooling was slower and uneven, with glycol-enhanced chilling drums. These rotating cylinders immerse the tuna in a constant vortex of chilled fluid, ensuring even distribution of temperature and eliminating thermal stratification. Over the following 12 months, lab tests showed a 27% reduction in enzymatic degradation markers. Clear evidence that molecular breakdown had been delayed. The difference could be tasted, seen, and measured. Not through marketing, but through microscope and market price alike. Because in this process, you're not preserving a fish, you're preserving a chain reaction, cascade of chemical responses that begin the moment oxygen stops circulating and blood stops flowing. You're pausing a metabolic process in mid-transition, halting the collapse of cellular membranes, freezing not just water, but time itself. Precision here is not decoration. It's the only language that muscle tissue understands. And when it's respected, the result is a product that still carries the ocean within it, a texture that resists the blade, a color that holds its promise, and a flavor that commands the highest bid. All because someone measured minutes, molecules, and temperature gradients like a scientist facing a deadline. Once the chilling phase is completed, the next stage begins. And it has nothing to do with speed. It has to do with form, pressure, and time. A 600-kilo tuna is not simply placed into a box. Its body, heavy and delicate, reacts to the smallest distortion during handling. If the load bends against an uneven surface, or if gravitational compression is not properly managed during transfer, what results is protein denaturation. At the microscopic level, fibers collapse, and tissue becomes unstable. What seems like a smooth transition from boat to crate can leave behind irreversible damage. Patches of soft texture, uneven slicing behavior, and a final product that no longer responds to the knife with the expected firmness. To mitigate these losses, some fleets have adopted vacuum cradle loaders, a technology adapted from high-precision fruit transport systems. Instead of hooks or mechanical arms, the tuna is supported by a semi-rigid membrane that adjusts to its shape and uses negative pressure to lift the mass without compression points. Attached to torsion limiters and flexible loading rails, these systems allow the fish to move as a single unit in harmony with gravity rather than against it. It's a method closer to handling a musical instrument than a commercial product, and it reflects a deeper understanding that fragility is not only on the surface, it's engineered into the structure of the meat itself. But even after the fish is secured in its container, the true challenge begins. Temperature must remain within a narrow corridor, typically between 1.0 wakes and 1.5 degree risk without oscillation. Surprisingly, slower shipments that maintain thermal stability often deliver better quality scores than faster routes with brief warming events. The reason lies in ice crystal formation. At the edge of freezing, a fluctuation of just 0.8 degree Celsius can initiate microcrystallization, forming shards that puncture cell walls and drain moisture from the inside. This is why the logistics system must prioritize thermal inertia, not just velocity. The most advanced carriers now use phase change panels embedded in crate walls, engineered to absorb excess heat without raising internal temperature. It's refrigeration through physics, not electricity. One of Japan's largest sushi restaurant chains discovered this the hard way. After noticing subtle declines in flavor and texture consistency, they traced the issue back to a customs delay at Narita Airport, a hold of just 3 hours, during which crates sat under ambient conditions, introduced a temperature rise of 1.3 degrees Celsius, barely perceptible in the data, but enough to trigger chemical instability. The chain responded by integrating AI-based route planners, capable of simulating thermal exposure along every logistical node and choosing pathways with fewer interruptions. It wasn't just about efficiency, it was about safeguarding molecular coherence across continents and through time zones, because in tuna transport, every stage is a negotiation with entropy, a continuous struggle to hold form against decay. When someone says, "every second counts," it's not metaphor, it's chemistry. Muscle structures is time-sensitive. Thermal consistency is not comfort, it's survival. And crate pressure is not just packaging, it's structural science. Every part of the process, from the curve of the cradle to the torque of the forklift, speaks a technical language, one that knows. The difference between a premium-grade fillet and a rejected load is not in the fish. It's in how the journey was engineered, measured, and executed, down to the degree, the angle, the minute. In the world of extreme food logistics, there is a fundamental shift taking place, not in speed, but in philosophy. When it comes to high-value species like bluefin tuna, the objective is no longer to simply ship a fish. It is to curate a biological event, because once the animal is removed from the sea, it doesn't become static. It enters a slow but irreversible process of molecular change. From that point forward, the challenge is not to freeze time, but to guide it with precision. And that requires more than a cold container. It requires a system that behaves like a clinical environment. Modern tuna logistics operates under principles far closer to biomedical handling than conventional transport. Each tuna is treated like a sample of living tissue under biochemical decay. And as with any medical sample, time, temperature, and handling method must be calibrated with exactness. What we're witnessing is the emergence of a new category of transport, one that draws more from pharmaceutical logistics and intensive care protocol than from the rules of freight forwarding. This mindset is visible in the tools, the monitoring systems, and especially in the design of traceability platforms. Japan's Tuna Track system is a case in point. It's not simply a supply chain tracker. It's a predictive model, powered by real-time data and thermal history that projects the expected degradation curve of each individual fish. From the moment a crate leaves the harbor, its internal conditions are logged and compared against optimal benchmarks. RFID tags carry not only identity codes, but embedded temperature logs and metabolic decay predictions. If a crate shows a thermal spike of even half a degree during transit, the system flags it for rerouting or reclassification. In effect, the logistics network behaves like a distributed ICU, with each crate as a monitored patient and each route as a treatment plan adjusted by algorithm. This level of control's not excessive, it's essential, because tuna flesh isn't stable after harvest. It is biologically active, undergoing enzymatic softening, water migration, and oxidative changes. The quality that arrives on a plate is not just the result of freshness. It's the cumulative expression of every decision made between capture and consumption, down to the choice of insulation, the curve of a transport hook, or the placement of a thermal buffer inside a container. There is no neutral moment in this journey. Every detail either protects cellular coherence or erodes it. And this is where the final insight emerges. The transport of tuna isn't a race against time. It's a negotiation with entropy, a delicate balancing act between biology and engineering, between nature's clock and humanity's precision. It mirrors other advanced agricultural systems, such as fertigation in vertical farming, where a delay in nutrient dosing can collapse an entire yield. The principle is the same. Life responds to timing, not just to care. The tuna arrives not simply because it moved fast, but because it moved smart. Each layer of protection, thermal, mechanical, algorithmic, worked in tandem to maintain what is essentially a living record of the sea. To move a tuna in 24 hours is to perform a kind of technical ballet, one that begins in salt water and ends under a knife, but whose true beauty lies in the orchestration between. A process built not on spectacle, but on respect for precision, systems, and silence. This is not transport, it is mastery, shaped by data, preserved by design, and expressed on a plate as something the ocean gave and engineering refused to waste.